All right, everybody. Welcome back to another video in the RNA sequencing data analysis kind of pipeline here. Today we're going to work with the GO and KEGG pathways. So previously we had gone through cleaning all of the data, and then we started looking at the data in R. We visualized the count file that we got output from feature counts. We went and did differential gene expression. We did some gene ontology things, which you can see we have left over from last time here. And then now today we can go on, we can look at the gene ontology pathways with KEGG pathways. And to do this, we'll need to import 3 packages that we installed at the very beginning. And that's going to be pathview, gage, and gageData. Pathview will let us view the pathways that we're looking at, and then the data that goes along with that. So the up and down regulation of particular genes within that pathway. And then gage is going to help us with the gene enrichment and the pathway analysis part. gageData is just an additional piece that goes along with gage, and that's got all the other like mapping and just extra stuff to go along with the gage package. So first, let's import those 3. So we'll do library, pathview, library, and then gage, and then finally library, and gageData. We'll run that. Let's add some blocks here. All right, now let's make an array for inputting into the gage function. We're gonna have to make an array of our genes log fold change along with the entrez ID for that particular fold change. So this will make more sense when we start looking at it. So if we do fold changes, we want to look at res, and inside res we'll do fold change. All right, so if we just look at fold change, now it should be an array of just the fold change values. It's not going to have any names that go along with it. So you can see just a bunch of log fold changes. We have to map those to the names, the entrez ID names, so we know what we're actually looking at. And to do that, we can do names, fold changes, so that array. And we want to give it the name of res and then the entrez ID that we mapped from our column name or our refseq IDs. So we can run that. And then if we do a head of our fold changes, we can see now that we have our log fold change along with the entrez ID. Now we have to bring in 2 kind of data sets from the gageData that are particular to the mouse data that we're looking at. And so for that, we can do data, and then we want go sets, and then for mouse, or musculus. And then additionally, we want to do go.subs.mm. We can run those, and it'll import it over here on the right-hand side. So we have go sets and go subs. All right. As an example, we can take a look at the biological processes. You can go through and do this for the other 2 Go terms, Go categories as well. But we'll just do biological processes here in the mouse data by selecting it out of the imported data that we looked at. So if we do go, and then we want to look at the biological processes, and then sets. So this is going to be our gene set, which we can use later in the gage function. So then we'll look at go sets.mm, and we want to pull out those which have an ID from subs biological processes. So if we do go subs $, and then we can choose biological processes, cellular component, or molecular function. So we'll just do biological process here. And additionally, now we can get our actual results from our Go biological processes by doing go BP, and then we can just call res like we did with the dseq, and then we'll use that gage function now. So first, the expressions here, it's going to be our fold changes. And then our G sets or gene sets is going to come from our go BP sets, which we just assigned. And we'll do gsets equals, and then up here we can do disperse. Great. Now, finally, we want to look at the same direction. So we'll call same.dir, and we'll set this equal to true. So what that's going to do is it's going to look in the same direction for up or down regulated genes. You can go ahead and run those. Take a little bit. What we can do after this is view our Go BP results, and we can look at greater than. So these are all the upregulated ones. Kind of hard to pull anything out, but a lot of morphogenesis happening. Let's look at less and see what we get there. So we have less messenger RNA processing, so that can make sense. Less translation, less DNA repairs, kind of harder to wrap your head around, but non-coding RNA, DNA recombination, RNA splicing, some interesting stuff happening. So if we go back and clear a lot of this out. All right, so save. Now that we have the Go part done, we can actually look at the KEGG pathways. And for this, we need to bring in 2 more data sets from the gageData package, and those are KEGG.sets, and then we'll want the musculus, and then additionally we'll do sigmet.idx.mm. So what the sigmet is it's basically I gotta rename this, smaller subset of the KEGG sets, which is only going to include the signaling and metabolic pathways. And for us, if we just want to look at that, we can do KEGG sets mm. So we'll subset our KEGG sets based on that sigmet. And then we can do KEGG sets.mm, so we'll look at the same package, but we're only going to pick out those that are in sigmet index.mm. We'll go ahead and run that. Now we can see that it was successful. Let's add a couple more here. All right. Now we can calculate the KEGG results like we did with the Go up above. So instead of using here G sets, so the gene sets of the Go BP, we can look at this new KEGG sets that we made. So we can call this KEGG results, and then we'll set that equal to gage, and we'll look at fold changes again. And then for G sets, instead of Go BP sets, let's look at our KEGG sets.mm, and then we'll do the same as before. We'll look at same dot direction. So we'll look at the up and down regulated. True. Okay. Let's open up the console again. And down here, just like we did before, we can go ahead and view our KEGG res, and we can look at greater than and less than. Oh, view. All right, so these are upregulated, and you can see that we have the hedgehog pathway. So that is going to be something that is attributed to if there is abnormal hedgehog signaling happening. A lot of times that can lead to the proliferation of cancers like pancreatic, lung, etc. Not that that's going to happen here if left undone or left unchecked because of the P53, but that is of note. What else do we see in here? There is P53 signaling pathway that is up. upregulated. We'll get to this in a little bit because there's something interesting happening here. But yeah, so that's pretty noteworthy, those two. Let's look at less than and see what we get. All right, so we have less DNA replication, cellular cycle is down, ribosome is down, RNA polymerase is down. So that's also interesting. You can dive further into that if wanted. You can Google like the MMU or the whole name, and it should give you a deeper explanation as to what exactly each one is. But now that that is done, we can pull up the actual pathways and start plotting them, and then we'll get some really cool pictures. And to do this, we won't need to, but we'll use some tidy piping, and we'll select just the top handful of genes and then produce some plots there too. So to do this, we'll do KEGG res pathways equals data.frame. And then the ID that we're going to pass in is going to be our KEGG res row names. So if we go back here, what we're going to do basically is create a list that just has the row names in them, and then we need to subset out this MMU and then the 5 numbers. So we're going to pick the first 8 characters that are in the names here, and that's going to be the ID that we can pull or put into the plotting path view. So the ID is going to equal the row names of KEGG res, and then we can do, we'll do greater than, and then KEGG res will just append on all of the data for the last part, less, greater. Okay, and then tidy can pipe. So we're gonna create that data frame, and then we're going to do tibble as tibble. So this is just something for tidy. And then we can filter. So what we're going to do now is we're just going to look at the top however many genes that show up in that table. So you can see here we have 177 entries. We can just pick the top, I don't know, 5 or so. So if we do filter and then row number less than or equal to 5, we'll just look at the top 5. Let's look at 20. Okay, so then we're going to pipe that, and then we want just the ID. So if you look here, we just want this name in that list, which is made here. So you do dot $ID, delete that one, and then we can pipe this to as.character. So we just want this list to be of characters. And go ahead and run that. Oh, I spelled it tibble wrong. There we go. And then we can just look at it. So let's look at the first, or however many in there. So there's 20. We can do KEGG res pathways. And there we are. So we have the first 20. So we have P53 here, and we can look at that. So dive deeper into that one. And then hedgehog signaling's in here. We've got the top 20 results. In order to get the particular IDs, like I was mentioning, these first 8 characters, we'll have to basically create a substring of these names here. And to do that, it's pretty easy. We can do KEGG res IDs. So these are just going to be the actual KEGG pathway IDs. And then we want to find the substring of KEGG res pathways. We'll start at the first character. So in R, the first character is a 1. If we were in Python, this would be 0. And then stop will be at 8. And then let's print this out too, so we can see what our new IDs look like. Whoops. There we go. So now we have just the first 8 characters for each one of these. Awesome. Okay. Now we can actually go ahead and plot them. So we can do tmp s apply, and then we'll look at the KEGG IDs, KEGG IDs, and then we need to write the little function, so PID, pathview, gene. So we need to look at the gene data, and that's going to be our fold changes. So it's going to tell pathview what to actually color our different genes within the path, within the plot. So we'll do fold changes. And then our pathway ID is going to be what's passed in. So we're going to pass in our KEGG res. So PID is just the function variable. So you can type PID here again. And then we need to specify what species this is in. Well, since we're working with mouse and the first 3 characters here are MMU, we'll do species equals MMU. And then if we run that, it'll take a little bit, but it's going to go ahead and it's going to build some plots. And if we go files, you can see possibly it puts them in here. There we go. Got some changes happening. And then we can pick out which ones we want to look at. So let's look at this MMU 04115. Let's look at the P53 pathway. 115. So something that I noticed instantly when I opened this up is that there is a lot that is upregulated. So the whole pathway is potentially upregulated, but P53 itself is actually showing a decrease. And I looked at the results table with the log fold change, and log fold change is actually negative, which then got me also looking at like what's actually happening there. So everything else but P53 is being upregulated, but P53, between the two, it's really kind of messy with your head, but let's just look at it to get an idea of how to explain it. So look at DDS, and then the gene. I already know what the gene name in the results or in the DDS is. So that's going to be our refseq ID that originally came with the data. So it's nm_00112723.1, and then int group. So this is basically what we're going to split our data by when we're going to plot it. And this has to be based on our column names. So call data, we have to pick something out of here, and we're going to look at P53 and treatment. So if we do P53, and then we also want to look at treatment, so it's going to basically parse this out into 4 different groups that we'll see. All right, so if we move this over. So the reason why it's negative fold change, even though in both the positive samples there seemed to be higher P53, even though in minus it should have been knocked out, is that because it's positive in both here, and the difference here is smaller than the difference here, in the P53 minus samples there was actually a greater change than in the P53 positive. So that's why when we look at the log fold change of results, so if we look at view every time, view as data frame, and then we want to look at res, we can search in here for TRP53. And this guy, we see that there's actually a negative log fold change and a pretty significant p-value. I mean, not incredibly significant, but it is significant normally, but not when it's corrected. So that could also be something, this isn't significant. But that's why when we look at that image, I gotta pull it up again. Here we go. P53 is negative, although everything else is positive. So there's one thing to just look at the data, but you got to kind of dive into it to get a better understanding of what's actually happening. So this is an explanation as to why PT3 in that picture is down, even though we have higher levels. So yeah, that should do it for the KEGG pathway. Oh, I closed it. Let's open it up again. That should do it for the KEGG pathway analysis part of it. And that should be it for the RNA sequencing as far as with the pipeline that we've been working with. So yeah, if you guys found any of these videos helpful, hit the thumbs up. That'll kind of give me a gauge as to if I need to do more of the R videos or if I should do more of the explaining the tools. I know the Trimmatic video has had a lot of interest, as well as the multi-QC and the fast QC video. Otherwise, feel free to subscribe. That'll kind of tell me how many people find them helpful and not just what exactly is helpful. But yeah, thanks for your time, guys.